Before we get started, if someone here is fan of the rock band Bad Religion, this title has an Easter egg. It's inspired by a name of an album of Bad Religion. So, if you're a fan, you will discover which is the name of the album. So let's go. So I'm Inácio. I'm postdoctoral researcher at Institut Curie. I work in computational biology, integrative genomics cancer team. It's a research group that works with brain tumors, pediatric and adult tumors. There are different projects regarding that. To analyze such kind of tumors, we employ analyze single cell or single nuclei RNA-seq and attack-seq data, basically RNA expression or chromatin accessibility data, as well as the next vision is spatial transcriptomics, which the area spatial transcriptomics is we are going to talk about today. But before, let's make a brief review of biology. So if you make a map between programming and biology, DNA is our Python code, RNA it's our assembly code, and the protein are the binary file. And right below in this picture, we see the process of compilation of such code. So basically, DNA it's replicated to form a complementary strand, then it's transcribed into RNA and translated into proteins. Roughly speaking, all of our cells of our body has the same DNA, the same Python code, the same sequence of nucleotides. However, which differs a neuron from an hepatic cell from our liver is how many times a gene is transcribed into DNA. So this rate of transcription, how many times it's transcribed, we call it gene expression. And over the last 34 years, what biology has done is to screen DNA sequences into computers to mount the genomes, so that, for example, in early 2000s we had the Human Genome Project. But besides sequencing and update the sequences of DNA, we also have RNAs, and here mRNAs, the RNAs that are translated properly into proteins, as well proteins. So biology has screened into computers lots of biological data here, and here we focus in RNAs, basically transcriptomics, because many of diseases like cancers and behaviors in plants and many other things are started with transcriptomics, analyzing gene expression data. But bioinformatics is much more broad than that, and there are other omics that you can study as well. Besides that, another important concept that we must review is the organization levels of biology, which you can see in this figure. Normally in bioinformatics we are focused in study the tissue, which is a midterm between the cells and the organs. So a tissue is a collection of cells in a structured way, and the organ will be the collection of tissues in a structured way. However, an organ can have more than one kind of tissues. So because of that, sometimes we focus on tissues. For example, when we study tumors, we study the tumor's tissue, its cell composition, and so on. However, besides this organization level into aggregating things, we also have spatial information. So here we have an image of the brain of mice, which in spatial transcriptomics is our iris dataset. So like in data science, we have the iris dataset, which the beginner's dataset in spatial transcriptomics is this one. And why? Because it's a very well structured organ which we can use to study biology and explore its areas. And given that the physiology of mice brain and human brain as well is well established and understood, one of the first questions that spatial transcriptomics aims to answer is how to characterize each region using gene expression. How can we say this region is different of this one because in this green region some genes are highly expressed compared to this other region and vice versa. So we try to answer that with spatial transcriptomics. The area, if you trace back to deep roots, it started in the between 60s and the 80s with in situ hybridization studies, but modern transcriptomics started overall with this paper from 2016, if I'm not mistaken, by a Swedish group led by Joachim Landenberg. And here we have Patrick Stahl, the first author. So they proposed a more modern way to capture not only DNA, RNA content, but also associated with spatial locations, such that with spatial transcriptomics, you know not only which is the sequence of DNA or RNA, but as well from which part, for example, from the brain mouse, that sequence comes from, as well as how is the expression here, how is the expression here in each point, let's say. So this was a revolutionary molecular biology technique such that in 2020, in Nature Methods, it was considered the method of the year because the unveiling of this spatial resolution of biological data made possible to formulate new questions and understand better how biology works. And in fact, biologically speaking, locations of things matters a lot. For example, not only in our brain, for example, neurons communicate between each other. So knowing which neurons are closer than others, or knowing which astrocytes are more closer to which neurons, help us to understand better the functioning of brain. Because for two cells communicate, changing signs between each other, localization is important. So how do we acquire this data? So a typical experiment for capturing is as we are seeing in the images. So our sample, in this case, for example, mouse brain, is put into a plate with little holes that we call spots. In each spot we have complementary DNA sequences, strands, from all the possible combinations with which the DNA is captured. And each spot, the information of XY coordinate of the spot is also informed. That's why we have not only the sequence information, but also the XY coordinate information. And this XY coordinate information is also encoded in a sequence tag associated with this probe. So all of this is condensed in a tube that is taken to a machine called sequencing machine to transform the strand of DNA into letters into computing. This methodology is called NGS based. There are other ones like in situ sequencing and in situ hybridization, in which instead of directly capturing the sequence itself, a process similar to taking a photo is taken, and based solely on the analysis of the pixels, it's possible to infer the associated the associated DNA sequence. The, the biggest difference between each other is that in situ sequencing and hybridization, you can also get the localization of each cell of the sample. And here in NGS based, this is not possible because each spot here of the lane comprises between 5 and 10 cells. On the other hand, in these two approaches, you need to specify which genes do you want to capture. While in NGS-based approaches, you can capture all the possible genes, all the possible genetic information. Once this information is captured, it passes for a computational process called alignment, in which the DNA sequences are compared to a database of genomes and genes to map each DNA sequence to its corresponding gene, such that our final output is a table like this, in which we measure how many sequences of each gene was collected in each spot of the lane. And besides the x and y coordinates of each spot. That's why we can reconstruct this image from the spots. It's not this slide, but it's in the next, that besides this gene information, genetic information, we also have staining information, hematosine, eosine information, and this is the basic data that we use for the downstream analysis. So there are many technologies for performing this kind of capture. The most popular one is 10x Vision workflow, which employs this spot-based capture methodology. And here we can see an example of capture bid, the structure. So here in the UMI and spatial barcode, we have the information about the X and Y coordinate, and also an identity code for each spot as well. We also have a poly-DT, which is a sequence of t-means that used to help to capture the read, the DNA sequence, and the read this DNA sequence itself. And besides the information, we also have the immunostaining, the immunostaining, but the staining, hematosine, and eosine also image, the histological image, which is also used for analysis as well. So here we have some of the outputs, examples of outputs that we obtain when you collect such kind of data for the next vision. The first one is the histological image, and here is a plot, as I've mentioned, the reconstructed image for the sequence, in which in each spot here we can see how many sequences of RNA were detected in each spot. And this is important for identify some bias and perform corrections, statistical corrections for downstream analysis. With this information of gene counts and gene expression, we can cluster the spots, initially only using gene expression data, not exactly spatial locations, despite recent works have been trying to aggregate spatial information to perform clustering, and thus we can get this nice structure. And interesting, this clustering made only from molecular data can reproduce the biological structures like that without taking into account spatial information yet. So it's a very striking advancement. So here is the next vision. We also have the next Xenium, which can get information per spot. So, and here we have some examples of spatial technologies and some examples of downstream analysis that can be done. Not only clustering, but identifies especially variable genes, cell-cell interactions, and so on. And how to analyze that? A popular Python library for that is Squidpy, developed by Giovanni Paglia from Thais Lab in Germany. It's a library that performs all of these kinds of analysis. And it is not only for visualization, but for also for computing spatial statistics. Because when you aggregate spatial information, we have to do a more specific and customized statistical analysis, because spatial data has its own particularities. So Squidpy has support for a wide range of spatial transcriptomics technologies. It's installable, and its base data is the gen per spot matrix data and also the histological image. Its base data structure is called annotated data in data that combines the original matrix with other matrices that have annotations for both spots and genes. So to do that, you just need to import the libraries. Here we import some available data sets that the library provides. If one line of code, spatial scatter, we also get some clustering structure for the image, but we get with this one line of code because the data we imported were already pre-processed and manually annotated each cluster. One example of statistic that you can do with spatial data is the Moran's eye statistic that takes into account the spatial location to perform correlation between genes. And it provides an index that goes from 0 to 1. The higher the index, the more spatially correlated the gene is with itself. And it is also a marker to say that that gene can be a marker for a specific region of your sample. So when we take these most significant genes and plot, we can see the many of them can recover specific regions from the sample. Another statistic that we can do is correlate regions to verify which regions has a similar molecular profile between each other. Besides working with vision, it also can work with Xenium, for example. And here we have an example of pre-processing that is normally made, which involves initially data normalization, PCA, data reduction, and UMAP to identify core features, and later clustering, clustering, which is a graph clustering, and later visualization. And here, because it's Xenium, it has not only information of location, but location of each cell. So here you can see a cluster of with higher resolution. Another technology that has high resolution is SlideSec v2. And another feature that Squidpy can provide is that you can just plot specific regions of interest. Because SlideSec and Xenium provide single cell resolution, another task that you can do with Squidpy is segmentation to identify the positions, the coordinates of the cell. So with spatial segment, you can visualize in more fine details the localization of cells, for example, using this data set of nanostring technologies. Currently, one of the biggest challenges in spatial transcriptomics is cell type deconvolution. Mainly for the next vision, because each spot in the next vision can comprise more than one cell, the task is which cell types are present in each spot. So many machine learning and statistical algorithms have been developed in the last years for that. It also presents big data challenges because it's terabytes of data, genomic information, and because knowing the distribution of cell types in the sample is one of the main questions of spatial transcriptomics studies in compositional data analysis. How to statistically study this are also being currently studied nowadays, and the proper study of spatial data analysis for constructing novel algorithms for understanding the distribution of genes. Besides Squidpy, there are also many other Python libraries for studying spatial transcriptomics, such as spatial data that provides a more refined and suitable structure for spatial data, stlearn for algorithms that pre-process and cluster data, you aggregating also spatial information, cell to location for doing cell type deconvolution, and NAPARI, which is a spatial transcriptomics visualization, which provides interactive visualization as well. So that's it. Thank you very much.
Well, thank you very much for your presentation. So we have time for questions. So please raise your hand if you have one.
I have a question, and sorry if you already said that I was a bit late. Thank you for your talk. By the way, it was really great. I have a bit of a cognitive science background, so this was really fun. We never got to look at anything with genomics. And my question was, you talked a lot about the data that gets mixed using the library, and I was wondering when the data about which cells are correlate to which region comes from. Maybe I was late, but I didn't remember you saying this. Where does this data come from?
Okay, where does data come from?
Yeah, because there are very different ways to do this, right? That you mentioned, and there is where does this data sets? I think it's a better question come from, where the correlation between a spatial region and the type of gene comes from.
Okay. So normally when you do the spatial transcriptomics sequencing, you extract the RNA of the sample, and besides the RNA information, you also get the XY coordinate. And with this gene information, you can map the information of gene expression to location. For cell type deconvolution task, there are publicly available single cell data sets, which is that data sets that has the expression of genes related to each possible cell type for thousands of individual cells. So in a cell type deconvolution task, you train a model with single cell data and use this model to predict the proportions of cell types in spatial transcriptomics data. So it's a supervised task. In my postdoc, I didn't have such data. So recently, there have been developed also unsupervised techniques based, for example, in independent component analysis that try to decompose the spatial signal into components and using this component information to map into gene signature database of gene signatures to try to extract some information. But the majority of the activities is using supervised techniques, and there's an ongoing effort to always have available single cell data appropriate for each spatial data that you have.
Thank you very much.
Thank you for the talk. Thank you for the presentation. My question is regarding the fact that in biological research, very often the great packages, great tools have been developed and published, but because of nature of academia, that people have very short contract. Basically, when a PhD student or postdoc finishes contract, it leave the lab, and you found in a couple of the years that tool is no longer maintained, and it's practically becomes more and harder to use it in the tasks. So my question is that this tools does it have like a community of support, or it's like lab who's supporting the and maintenance of the tool, and what is currently how it's maintained in and how in biology it can be addressed this type of issues.
Yes, this is a valid and real issue with the academic world, but in fact, most of these libraries have a supporting community. Inclusive, Squidpy is a package that is part of an ecosystem of tools called SCverse, which is a collaborative network of labs, academic labs around the world that help to develop these tools. So yeah, in this area, there's a big support of community, and each year, many packages are always released, and at least in Curie and in other institutes, there's this emerging spirit of continuously maintaining the tools. It's something that I feel that's changing a bit.
Thank you.
Are there other questions? Otherwise, I have one, but maybe when I ask this question, is the next speaker in the room? Not yet. Hendrik? No? Okay, but I give you the microphone.
Thank you very much for the talk. I'm actually curious, it's not exactly related to the talk that you've given, but what do you think of foundation models? Like, we had a lot of talks today in this room devoted to LLMs. What do you think of the equivalent for transcriptomics data, something like single-cell GPT and other models like that? Are they useful at this stage, or do they have potential to be used in the future?
Yes. Foundation models and large language models are AI technologies that have been increasingly being adopted in the bioinformatics community. Regarding spatial transcriptomics, as far as I know, there isn't yet LLM applications, but for single-cell technologies, there's a lot of work. One of them is that one mentioned, SCGPT. It's a foundation model for single-cell data, and it can be used not only for doing cell-type deconvolution, but also for other tasks regarding single-cell data. And yeah, not only there's much room for applications in spatial transcriptomics, but also I think that sooner or later that there will be some either foundation model or LLM package for spatial transcriptomics as well. The main challenge here is that each sample have a different format, a different shape. So it's a very heterogeneous thing, but the one who can map this into a large language model or in a graph neural network will be very successful in that.
Thanks.
Thank you very much. So let's thank the speaker again.